Okay, gang, we need to perhaps return to everybody somewhat talking to one another. I know that it's a long list of questions, and we really haven't had time for, I think, any subgroup to go through it, but perhaps if we could start over here and have each table in turn appoint somebody at the table to come up with mention a couple of the points that they felt were most salient out of this list of topics that are questions from yesterday we distributed. Chuck? Okay. Somebody at Katie and Chuck and Jean and Vivian's table.
Okay, I'm going to speak because I was the most difficult one to convince or explain things to. So the first question is, what aspects of the built environment shape the microbiome environment? So we feel we have to get the fundamental question on the table so we know what projects. And then the second question to that is, okay, so we now know which aspects of the built environment shape, and the word shape was carefully selected because it's not just to sponsor or to diminish, but actually, if the physical environment can in fact change it, we need to know exactly what we can do in the physical environment to shape the microbiome environment. As soon as we talk about that, we need to know what aspects of the built environment microbiome impacts human health. And we spent some time trying to distinguish between built environment microbiomes and human microbiomes, since that was raised yesterday as a distinction. And I'm going to try this definition. I think we've got, I don't know, this is hard because I'm a built environment person and everybody else at the table knows a lot about microorganisms. So the built environment microbiome is the collection of microorganisms in the built environment that reside in or on the water, air, surfaces of buildings, and we don't know if we're going to say in their immediate outdoors. So we've defined the built an indoor environment, really, with significant influences from humans and other living transport mechanisms. So there's some interaction effect that we haven't figured out. The human microbiome is the collection of microorganisms in the respiratory, skin, and digestive systems, and any other human system that might be supporting them. So we're trying to parse those two out. And then the question is, what is the interaction of the built environment microbiomes and human microbiomes, and how do they affect human health and possibly other outcomes? We hadn't quite decided if we were going to only focus on human health, but so leave some other room on the table. And then what are the key adverse and beneficial outcomes for humans caused by built environment microbiomes? I think we spent a lot of time trying to make sure we knew what we were going to write these questions around.
Okay. No, and thank you. And you're going to send us these wonderful definitions?
Yes. Yes.
Thank you. Susan?
Could I just add a clarification that microbiomes are microbes and their genomes. A microbiota is a profile of the microbes who are there, and it's a very clear distinction that we're trying to make sure is propagated in the literature. So microbes and their genomes for a microbiome.
And where in the literature are you propagating this distinction? No, I had not seen those stipulated definitions, and I'm just curious. And where is it, the official definition?
We're writing a piece for one of the journals that I edit to try and clarify this. So microbiome is trying to clarify these definitions.
Okay,
there's some
and
position
so
papers.
if we could have
Sure,
have
I can
that
source
would be,
that. Sure.
Oh.
I'm so sorry, you're running back. You're getting fit this morning. So with sequencing, 16S sequencing, for example, you can generate a profile of which microbes are there. So you know by just a biomarker gene which microbes are there. But microbes are not just 16S, they come with a genome. So we think in terms of a microbiome as the organisms, their genes, and how they interact with each other and their environment, as opposed to just simply a cataloging approach, which is a microbiota. Excuse me.
So you're sort of making it
their
a
functions.
flora and fauna is the microbiota, the listing of what's there. Is that the
The
distinction?
list of what's there, the catalog of what's there is a microbiota. The microbes that are there and their functional capacity and how they interact with each other and their environment is a microbiome.
Okay. Thank you.
So I know the definition very confused. So I read some papers before. I think so. The microbiome is a collection of genes and organisms. So then this micro community, all the equivalent to microbiota, is just like a collection of organisms. Yes, you can use 16S because for bacteria it's easier because we use 16S, the same thing with organisms. But if we treat it as like micro eukaryotes, we don't use molecular markers. We just use morphology. So then this will be easy to understand. So I think the microbiome should be included both organisms and the genes, the collection of genes and organisms. Yes. Yes. So the microbiota just the top of the organisms. But organisms, how to define the organism? For bacteria, you can use the 16S. For others, eukaryotes, you can use morphology, any other tools.
Yeah, let us perhaps revisit the distinctions of etymology at another point, because I'd like to get each table to go ahead. And so Andy or Mark or Ron or somebody at this table.
Okay, so I was elected. So one of the first things we talked about was trying to get a practical understanding of what is a normal microbiome for a building, and that way we could learn about how that might affect the building materials that might be used or implemented in making design choices. Another thing that we discussed was what kind of monitoring systems are currently online or need developing that we could track microbes in a building, sort of as indicators or sensors in a building. And then one of the other topics we discussed was if the answer on the question number 1 on our sheets about the health effects of buildings are answered, once we get more information on that aspect, then we can talk about implementing design techniques that might address those questions. But first, I think we need to know more about what are the actual health effects. Is there anything I missed?
I just want to add a comment. Over my career, I've had experience in the design and assembly and operation of engineering systems of a number of different kinds. For example, in the case of power plants, selecting the materials of construction for a power plant is today pretty prescriptive, because we know a lot about the fact that there are certain elements in the environment, the working environment, the service environment, that can cause problems. Mm-hmm. For example, if you're building a steam generator for a nuclear power station, you must avoid exposure of those materials to halides, because halides will cause cracking of the steam generator tubes. So what do? You install monitors that will detect an immediate intrusion of species like chlorides into the environment, and you shut the plant down until you can control it. Okay. Now, from a 30,000-foot level, that doesn't sound too different from what we're talking about here. Mm-hmm. I mean, I did learn a lot yesterday, although I would say what I learned mostly is a very complicated issue, the biome. That's probably the understatement of the week. But I think if we understood a little bit better which of the multiplicity of species that are present in a building biome, and we knew which of those were of greatest concern, it may be possible to produce monitors and sensors that would detect them, and then respond to that. And I mean, that may be decades in the future, but the fact is, I think it might be at least a formalism that would be worth thinking about.
Thank you. Very, very good point. Okay. maybe we can move to table 3, arbitrarily, table 3.
Yeah.
David?
Okay, our table spoke about a number of things. We were trying to rationalize the questions and also maybe ask about some of the things that might be underlying some of the questions. It noted that a number of the questions can be summarized in the sort of meta question, what is the relationship between humans and MOBE, and more specifically, how does exposure to MOBE affect the human microbiome and health outcomes? Who is most susceptible, when in their lifetime, and why? One of the points that was brought up that wasn't a part of any of the key research questions was, what are the standards of proof that should be applied before making any recommendations? What should we be looking at to validate our findings before we feel confident enough to draw conclusions, to make recommendations? Another thing that we spoke about was how, given how difficult it is to really get a handle on this thing, to ask ourselves in greater detail, what do the natural experiments tell us? Unfortunately, we didn't have Jack here today. I know that he has had some experience in the hospital environment, which we figured was a controlled enough environment, and an environment where there is active attempts to manipulate the microbiome. Then what can that particular environment tell us? What can the natural experiments that are happening every day when people do cleaning in those environments
Did you come
tell
up with
us?
other examples of natural experiments, because like the hospital, the cleaning, others?
We didn't. I'm sure others exist. It just seemed to be a particularly controlled and well-controlled environment. I know with my own experience in getting things past IRBs, I have difficulty imagining circumstances where anything other than natural experiments would ever pass IRB approval. So it may be that
I was thinking of NASA's interest in this and some things.
Certainly in, well, submarines, the space station are two other places where those kind of experiments are going on.
What would be the hypothesis of the natural experiment? I mean, what would you be doing the natural experiment to find out?
Under what circumstances?
Yeah, it would be to, well, why don't...
Sure. So the question was, under what circumstances can we or should we manipulate the microbiome of the built environment? So under what circumstances, to what end? And we thought that we would be looking at natural experiments where people are already doing that manipulation to understand when it would be a good idea and when it might not be a good idea.
For example, one of the things we talked about was elder care or nursing homes. So, so you said there was one study.
There's a study from the Breakout Fork in Ireland. It's fine.
All right. So comparing those who convalesce or age at home versus in a home. So one would be highly manipulated. The other would be a quote natural environment. So something like that.
Okay, yeah.
Yes. Right.
Okay.
Oh wait, one more. Built. And then the impact on the human health. So that would be the. So one thing that seems we need to articulate is that we're really talking about three actors in this, and that would be the human, the microbial environment, and then also the built environment, and that these are three separate things that come together. So the human, the microbial environment, that would be, for example, in the built environment, because that's the context we've been talking about, but then the built environment itself, so that's the construction of that, so the piping, the HVAC system, the materials that the building is made out of, that is separate, and it seems to also be talked about because it can degrade, it can facilitate. Yeah, 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 yeah. Yeah, because we talk about them, but we don't articulate that this is something separate as well.
Yeah, we're definitely circling around the definition point. I think Diane wants to say something.
Just a small example of a natural experiment that Gary Adamkowitz brought up, which is the green building new housing in disadvantaged neighborhoods, housing that can be, quote, green and tighter, and people move from one type of housing to the other.
Yeah, good point. Okay, let's hear from table 4. Do you want to, Jessica, do you want, and then we'll do the last group with Jeff last. Okay. Oh, well, Hal wants to go? Okay, Hal, go Hal. Yeah, if you want to. I mean, I thought that's what you were saying, that you wanted to go. Okay.
Okay, all right.
Okay.
So our group, a lot of what people have already said resonates with what our group discussed. That we wanted to have a broader overarching question of, does the built environment microbiome affect health outcomes? And by health outcomes, we talked about human health, public health, and also building health. And we made a point at this table to make sure that that included, from a human and public health perspective, susceptible populations. And we learned a little bit about the definition of what a susceptible, what a susceptible population is. The second question is, we did really like the question, are we looking at the right things? We realized that this is really a methods-based question. Do we have the right sample designs? And are we getting the right information to be able to answer the first question with regards to how are we going to get information about the virome, about more information about the function of microbial communities and eukaryotes in the built environment? We also like the question, can and should we manipulate the built environment microbiome as it pertains to the last two questions that we thought were important? One is, can we optimize building and city design to proactively respond to what we called megatrends? And two megatrends that we talked about were increased population densities in cities that are coming over the next decades, and also climate change. And the fifth question was, can we mathematically model built environment microbiomes in a more rigorous way, similar to what's being done in physics and chemistry, so that we can respond to all the questions that we outlined before that?
Thank you, Jessica. Okay. We won't forget you, Hal. Thank you.
Okay, so I think we had 3 major themes that I did not manage to capture in incredible detail. I think 2 were roughly similar to what was already discussed, and then there was a third on climate change. So what impact will climate change have on indoor microbiomes? How do we track that? Does it matter? How does it affect human health? And then we had, what is in the indoor microbiome and how does it impact human health? I think that's a similar theme, right? And what are the practical implications for building operations, et cetera? And does operating a building for good indoor air quality using best practices promote positive health outcomes for people and the indoor microbiomes, or perhaps that's a negative for the indoor microbiome, but good for people, depending on how you look at it?
We also, harking back to Brent's slides of yesterday, where he had sort of a typical dry building biome and a typical wet building biome, we thought it would be nice to fill that out more with a systematic study of the biomes of buildings that are known to have been dry for a long time and buildings that are known to have moisture problems.
Hal.
So this was mostly distillation of the questions that I'm interested in. And so the first one is to study the interactome, microbes, and the built environment, including the chemicals on the surfaces or the air where the sample is taken, and to do that with attention to the dynamic processes that go on. So a single grab sample would not capture the dynamic processes. We need to study the changes that go on in buildings and within buildings over time, and look at that in relationship to human health and the building structure. A lot of this has already been brought up by others. So that combination, study the interactome and look at the human health and the impact on the building, is the top priority. The second one is a round-robin lab study, looking at the methods, getting to the species level, and looking at the effect of different labs' processes, including the presentation yesterday we heard about the difference that the primers make. The third one was brought up yesterday, to do an analysis of the burden of disease in the U.S., and I listed 5 adverse health outcomes that various research have looked at: asthma, respiratory infection. So you've got 15% and rising rate of asthma in the population, respiratory infection, commonly reported as 2 infections per person per year, cancer, allergy, and skin diseases, and I'm sure I'm missing something that.
How about neurological problems?
What's
There's
that?
Neurological problems? There's a
Yeah.
lot of smoke around that.
Yeah. So there needs to be a burden of disease analysis, and then even after you have the data, I think different people are going to. I've been thinking and writing about this for a long time, are going to rank the different health outcomes differently. And so how do you compare lifelong asthma with the possibility of cancer or early death? How do you compare having severe allergies to neurological problems, for example? And these get into values, and there's very little literature about this, most of it around trying to value death for engineering, like highway design or earthquake hazard mitigation. And I think this began with Love Canal, where doctors were worth $1 million and teachers were worth 100,000, and they needed to prioritize where they cleaned up the Love Canal disaster. And it was done on the basis of projected earnings over the remainder of the life, so age came into the play. That's one approach, which is dollars. But if you're an asthmatic, or if you're the parent of an asthmatic, the costs aren't quantifiable only in dollars. It affects quality of life, and you might rank those differently. So that's an important challenge that I think underlies where you would prioritize what you would study. And in the Sloan community, I hear often people talking about either bacteria or fungi, and others in the room hear it in terms of whether they study bacteria or fungi, as opposed to being able to understand that the comment was limited to one or the other, and then the omission of viruses, which is common but unacceptable. So.
Okay. Joe. Leanne?
I have a practical question. What is the infrastructure planned for all of this to happen? I mean, is there an idea for what?
What do you mean by infrastructure?
So in nanotechnology, right, there's the nanotechnology national infrastructure funding system, for example, where you know, there's sort of a national office, and then there's satellites at various universities, and so there's communication among the universities with the national office. And so is there a picture of some kind of infrastructure that it would allow all of this research to happen with some kind of root system that keeps everyone connected?
I think yesterday you may have seen what the issue is. There are many existing agencies, and then Sloan Foundation, which is wrapping up, have been throwing energy and grant dollars and whatever, and that one reason for this study is an attempt to add on to the coordination. And you heard that the White House report has also done, they brought in money from a couple of new foundations, but no, there's no government agency right now that owns this particular problem. And that's often the issue with interdisciplinary studies, where you find that people have been looking at the same thing from different angles. And so one of our goals, you know, in terms of the product of this National Academy study, is we've been charged with identifying some of the burning issues that agencies like EPA and NIH, should they choose to get more involved, can plan ahead in their strategic planning. Yes, Vivian?
Just a note of caution, the built environment has no natural federal funding stream to look at quality degradation. National Science Foundation has consistently argued that the built environment is an applied field, not a fundamental science, and not meriting its own division of research. The Department of Energy, of which 40% of the energy used is inside buildings, puts about 4% of their total research budget into the built environment, most of that to national labs. HUD is probably, in terms of housing, one of the best sources, but they too have an incredibly delimited amount of. So I think when you talk about the infrastructure, the first thing is to convince federal agencies that the built environment is worthy of research funding, and especially collaborative research across disciplines, which is really a key issue. So just, there is no champion right now.
Yeah, and the Sloan Foundation has been a champion, having identified this as an area that needs help. And their goal, you know, written into the Sloan plan, is that they spark something, and then they want it to be turned over to the federal funding agencies. And I have not been part of this MOBI. Maybe Jeff, you may want to speak to that long-term strategy of the Sloan Foundation and how it's wrapping up, or someone else, Jessica, who's been Sloan funded.
Yeah, I mean, I think it's an issue. I think that it's a huge issue. That I don't think that there's a good plan for going forth. But the built environment has always had this kind of tough issue of funding, of the applied versus fundamental. I mean, it's not just MOBI, it's everything. And that's the history. And there's always been encouraging signs, I think. HUD, which was just mentioned, in Canada, our equivalent of NIH has opened up a stream in the built environment for possible funding. So I think there's possibility of that, but I don't think that there is a framework in place right now.
Okay. We're sort of running over the time for our given break, and we do have some formal presentations up. So burning questions, or otherwise I suggest we take our break now, and then we're scheduled to reconvene at 10. How about 10:05?